I've uploaded to insect synthesis phylogeny of insects, 28,000 species. Which comprises the machine-readable phylogeny here and a precisely matching file of co-1 DNA barcodes, so each same 28,000. So to build this synthesis, I've been going through order by order to try and scrutinize each subtree and reduce the major errors. A synthesis phylogeny will always have certainly more errors, perhaps even a higher error rate than a taxon-specific tree due to various things. But of course, better to reduce these. So completed all the orders apart from there's 3 orders which are either tiny or they have negligible public sequence data. So both completed most of the orders, and I've just finished stitching these all together into a single synthesis. So to compile these required so here's the relationships between the orders. It says in insects is about 29 or 30. Neopterida is actually 3 orders, and there's one order called Isopter which are included in Blatodia. That said, and a single outgroup. But here's relationships between the orders according to Mishoff et al. So for each of these, it requires retrieving results. So I started with Lepidoptera. This is one I've spent a particular lot of time on due to various things. It has a lot of barcode data, and we study these a lot. So the tree is copied into the working directory. Now each of these order syntheses has, if you're going to put it onto a backbone, here you have to, this is the synthesis for this order comprises the in-group and a single out-group, so you have to remove the root and the out-group of each. That is a main task, and that's done here. So in this case, the out-group, which is my crop turner, is at the start of the file, and the end of the file has a branch length on the root, although the length is 0. So there's several different configurations. We have the outgroup at the start of the nuic file, and at the end we have a branch length on the root, or we can have the outgroup at the end of the file and a root without branch length, or we can have the outgroup at the end of the file and a root with branch length. And there's one other I came across, which I think it was for Coleoptera, outgroup at the start of the file and root without branch length. So these are all different configurations, but all produced by RaxML for whatever reason, but all these are rooted. So as I say, you've got to just remove the branch length, remove the outgroup, the root branch, and the root branch length if present. So a replacement regular expression is needed for the different configurations, and that's done for each of the order level syntheses. And you get these tiny insect orders or those without any significant public sequence data, Zoopto, Mantophasmatoidea and Grillobatoidea. So these are just, I did found a single barcode, and I just represented that each of these with a single sequence, and we have one diploid outgroup. So these are all then also retrieving the FASTA colon FASTA files for each, and producing an exactly matching file. So although I've removed the outgroup, I can't simply remove the corresponding co1 from the data, because that may actually be included in the in-group or a separate one. So there's that to consider. And finally, writing so I should say, once the outgroup's been removed, we then replace that order name with the order level synthesis, so these are all replaced with these subtrees, and we end up with a synthesis. I then simply recalculated the branch lengths to make the synthesis, and here's the result. You can scan this in Archaeopteryx. There's not many other software which will load this size, and so you can see the terminal set. And now most syntheses are, apart from highly studied things like birds and mammals, so for most insects, when there's been an attempt at a very large, you get very messy taxonomic structure of the set of terminals, because of if synthesis is made from public sequence data, because you get various things unlabeled, and species repeats, and non-canonical things, and all of these sort of add up to divergence between the number of terminals and the number of species, such that you're not even sure what you end up with. But you can see here I have precisely structured the set of terminals such that each terminal is a single unique species. Now this is efficient. I mean, you could have non-canonical things in there, but there's not much you can do with them. Like, you can't attach them to biological data, for example, so they're not much use as a reference system. So now I prefer to have this very neat species level structure, and this is a lot more efficient. And this is now available, and the next job is trying to increase the number of species, of course. So the quality of this should be now a lot higher than previous syntheses, but it'd be nice to have a lot more species. I'm currently working on ways to graft in extra things.